Good afternoon, my name is Simar Arora, and today I will be presenting research on the effect of insect repellent and UV radiation on the embryonic development of DANIO RERIO. The purpose of this experiment was to examine the effects of environmental toxins on developmental stages of zebrafish. This was done by examining two stages of development, 24 and 48 hours after fertilization. In this experiment, insect repellent was used as a substitute of insecticide so that the embryos were not completely eliminated, but the effects were still mimicked on a smaller scale. The two environmental toxins chosen were UV radiation and insecticide. Ultraviolet radiation is a form of electromagnetic radiation emitted from the sun. It has been associated with damage to plant and marine ecosystems. Studies have concluded that UV irradiation affects the photosynthetic abilities, metabolic rates, and motility of aquatic organisms. It has also been found that exposure to solar UV results in embryonic mortality in amphibians. Despite these harmful effects, Earth has historically been protected from UV rays in the sun because of the ozone layer that encompasses Earth. In recent times, however, the ozone layer has been depleting as a result of the use of chlorofluorocarbon gases, CFCs, which break down the ozone layer by releasing excess chlorine into the atmosphere. Exposure to toxic chemicals as a result of biological accumulation is also a harmful environmental factor. Biomagnification is the increasing concentration of a toxic substance as it moves up the food chain. Toxic substances include pesticides, insecticides, metals, etc. One major example of biomagnification can be observed in the chemical DDT. DDT was developed in the 1940s as the first modern insecticide. Farmers used DDT, and the chemical runoff was unintentionally released into bodies of water. From there, the DDT began affecting the phytoplankton at the bottom of the food chain, and as predators consumed these organisms, biomagnification occurred, resulting in a higher concentration of DDT in larger fish and ultimately causing widespread decrease in marine life. Other insecticides have also been proven harmful to the biosphere. Pyrethroids, for example, are often used in insecticides and have been proven to be harmful to aquatic ecosystems. In 1991 and 1995, deltamethrin, a type of pyrethroid, caused widespread devastation of eels in Lake Baltan, Hungary. These examples prove that biomagnification of insecticides have detrimental effects on aquatic ecosystem. That being said, it is clear that UV radiation and biomagnification of toxic chemicals have increasing environmental significance. The embryonic development of zebrafish has many different stages, including the cleavage stage, the blastula stage, gastrulation, organogenesis, and hatching. This experiment mainly focuses on an important stage in gastrulation called epiboly. Gastrulation is a period of embryonic development in which cells migrate to form the major axes and germ layers of the embryo. Epiboly is when a layer of epithelial cells spread across the surface of the blastula, or mass of cells of the embryo. Since zebrafish embryos are transparent, epiboly is notable on the embryo when viewed under a microscope. Another part of development where data was collected in this experiment is the long peck stage, 48 hours after fertilization, when the embryo is just about ready to hatch. At this stage, the heart rates of the embryos were taken. Knowing this background on the development of zebrafish, one more important point of data to know about the effect of radiation on zebrafish development is that in a study by Alcaraz in 2001, UV radiation was found to cause a high occurrence of mutations in the epiboly stage of gastrulation. In this experiment, insect repellent was used as a substitute for insecticide. Embryos developed in different concentrations of off-deep woods bug spray. Off-deep woods bug spray is an insect repellent with the active ingredient DEET. It has been demonstrated that DEET has low to medium toxicity on G. afnis, or the mosquito fish, and resulted in disorientation on fish. So for the purpose of this experiment, DEET would be more effective than a chemical with high toxicity, which would probably eliminate all zebrafish embryos. Since UV radiation is a universal carcinogen, it was hypothesized that UV radiation would hinder embryos from developing to 50% epiboly more than insect repellent would. Additionally, because carbaryl, a type of insecticide, was found to cause induced bradycardia by C.C. Lin et al. In 2007, it was hypothesized that insect repellent would cause lower heart rates than UV radiation. The materials used in this experiment were the following: a fish tank, zebrafish embryos, zebrafish, marbles, UV light source, insect repellent, spring water, pipette aid, serological pipette, disposable pipettes, Petri plates, and an inverted microscope. Before experimentation, different concentrations of insect repellent were prepared: 0.01%, 0.1%, and 1%. In order to collect zebrafish embryos, the embryos had to be separated from the adult fish. Zebrafish tanks were filled with marbles so embryos could accumulate at the bottom of the tank. Embryos were then pipetted out of the tank using a serological pipette and pipette aid. Embryos were transferred into petri plates with 15 milliliters of spring water or prepared concentration, using a disposable pipette. For the insect repellent plates, no further preparation had to be done, and the embryos were allowed to develop. For the UV radiation trials, the embryos were put under low-power UV light for different exposure times: 15 minutes, 30 minutes, or 1 hour. The next day, embryos were checked for epiboly, and the day after that, heart rates were taken. T-tests were calculated between each experimental group's heart rates and the negative control of no treatment. These tables show percentages of embryos to reach 50% epiboly in different treatment groups and at different intensities. Data was collected 24 hours into development. Insect repellent has an overall higher effect on epiboly, with an overall of 50% of embryos to reach epiboly, while UV radiation had a slightly higher 53.66% of embryos to reach epiboly. The data can be better visualized on this figure. It is important to note that none of the embryos in 1% insect repellent reached 50% epiboly. Embryos at 0.01% and 0.1% insect repellent concentrations had consistently higher percentages of embryos to reach epiboly than UV treatments. Therefore, it can be concluded that insect repellent at a higher concentration has a greater effect on epiboly than UV radiation for a longer exposure time. This table shows the average heart rates of different treatment groups. No data for heart rate of 1% concentration treatment could be collected, but both 0.01% and 0.1% insect repellent demonstrated significant data against the control of no treatment. Heart rates of embryos exposed to different exposure times of radiation also all demonstrated significance against the negative control. The treatment group with the lowest p-value and the lowest average heart rate is insect repellent at a concentration of 0.1%. Considering 0.1% insect repellent was an intermediate level of exposure and showed more significance than the highest level of exposure to UV radiation, it is reasonable to conclude that insect repellent has more of an effect on the heart rate of embryos than UV radiation does. This figure is simply a graphical representation of average heart rates of embryos at different concentrations of insect repellent. 1% insect repellent is not included because all embryos were eliminated as a result of the chemical. The asterisk indicates a significant difference between the data and the negative control of no treatment. And as you can see, both treatment groups here had a significant difference. This figure is similar to the previous, but is for the UV exposure treatments. All three treatment groups had a significant difference compared to the negative control, but between each different time period, you can see that there is a lot of overlap in the standard deviation bars. Based on these results, the hypothesis for this experiment must be rejected regarding epiboly and accepted regarding heart rate. It was hypothesized that UV radiation would result in a lower percentage of embryos to reach 50% epiboly because of its classification as a universal carcinogen and its various effects on different organisms. The results of this experiment demonstrate that although UV radiation did hinder embryonic development in a way that 53.66% of embryos reached 50% epiboly, insect repellent stopped the development of the embryos slightly more, with only 50% of embryos reaching 50% epiboly. It was also hypothesized that insect repellent would decrease heart rate more than UV radiation because it has been suggested that insecticides result in bradycardia of zebrafish. This part of the hypothesis is accepted because 0.1% insect repellent resulted in the lowest average heart rates of embryos and the lowest p-value when compared to the negative control, meaning these data are the most statistically significant. This could be because UV radiation is constantly emitted by the sun, while insect repellent is an artificial toxin. The embryos might not have any defense mechanism against chemicals like DEET. The error in this experiment consists of inconsistent UV radiation. One trial of UV exposure was done with a higher power setting than the other trials. This could have resulted in a percentage of embryos to reach 50% epiboly that is slightly skewed down. This could be remediated with more trials. Another error present is that it is impossible for all embryos to be at the exact same stage of development when treated with their respective treatment groups. So some embryos may have been closer to 50% epiboly than others, resulting in random error. Because of time constraints, only 3 trials per treatment group, each with about 5 embryos, could be conducted. For more accurate data, more trials with more embryos should have been conducted. The data indicate that increasing exposure time of UV radiation does not have much of an effect at this degree. This is shown by the large overlaps of standard deviation and the small difference between the average heart rates and percent of embryos to reach 50% epiboly. This could be because UV radiation is constantly emitted by the sun and exposure is continual in real life. That being said, one possible extension of this experiment is to determine at what exposure intensity and amount of time UV radiation becomes as toxic as insect repellent, or when radiation becomes too much for the embryos to handle. If more resources were available, another extension could be to determine what genes are being affected by these environmental factors that cause the hindrance in development. Thank you so much for your time. I hope you and your families are staying safe in light of everything that's going on. Thank you.